Hello, my name is Luca PEZZAROSSA from the Technical University of Denmark, and today I'm going to present the paper titled A Computer Vision Algorithm for the Digitalization of Colorimetric Lateral Flow Assay Readouts, which I co-authored together with Susan Preus, Vinnie Svensson, and Jan Madsen. This work is part of a bigger project which aim is to develop a cheap and quick test to detect the presence and concentration of bacteria in wounds. Here you see a rendering of the sampling device, which shows the concentration of certain strains of bacteria as a color marker. In order to have a precise measure, we have a digitalization process which extracts some digital data which can then be used for analysis, visualization, and storage. In this paper, we focus only on the digitalization part, which is carried out using a computer vision algorithm. This is the outline of the presentation. I will first give some quick background on lateral flow assay and colorimetry, just to put the work into context. Then I will go through the explanation of the digitalization algorithm, which is the main contribution of this work. Then the experimental evaluation, and finally the summary of what we presented. Let's start with the first point. Here we see our sampling device, which is based on lateral flow assay. A lateral flow assay is essentially a strip of paper which can drag liquid due to capillary forces from one side to the other. A typical lateral flow assay consists of 4 regions. The first one is where you put the sample, then we have 2 regions where the reaction happens, and then finally an absorption pad which collects all the liquid at the end. Now, since in our case we want to detect bacteria, on the conjugation pad we're going to put antibodies for those bacteria which are conjugated, which are attached with gold nanoparticles. These nanoparticles stain the bacteria with a certain color, in this case red. In the next region, in the reaction membrane, we attach the antibodies to the strip. They are immobilized so that they can capture the bacteria. In this case, we have antibodies able to detect 4 bacteria. Let's see how all of these work. We put our sample with the bacteria inside, which here are shown as black spots. The sample starts moving, dragged by the capillary forces, and bacteria are getting stained with the gold nanoparticles because the antibodies attach on the surface of the bacteria. Everything moves forward, and the bacteria are then captured by the antibodies that it is immobilized on the strip of paper, therefore forming a certain color spot there, a color marker. If we look at this as a real image, we can see the spot. Now, these are real images, and we can see that they are different intensities. And this intensity of the color of the spot is correlated to the concentration. And this is part where colorimetry enters into the work. Colorimetry is the relation between the color and the concentration. So there is a certain function that correlates the concentration of the bacteria with the color. Therefore, if we can detect precisely what is the color and we are able to distinguish between little variation of the color, we can say quite precisely what is the concentration of the bacteria that generated the spot. Lateral flow assay, together with colorimetry, have several advantages. They are cheap to produce and provide quick response, making them very suitable for point-of-care applications. Colorimetry allows a quantitative measurement of the analyte concentration, in this case the amount of bacteria. However, the lab equipment for colorimetry can be expensive and it is quite bulky. The current colorimetry techniques rely on the alignment of the color marker. This means that the position, shape, and size of the marker is known in advance. In this work, we want to remove this constraint and be able to detect the marker, his shape, his position, and his size, and finally average his color without knowing in advance where it is. Now we move to the main contribution of the work, which is the digitalization algorithm. Just to summarize, here you can see the steps. At first, the LFA, lateral flow assay test, is performed. A picture of it is taken. Then we have our algorithm running on a computer-based system, and we get out a digital measurement, which, as said at the beginning, can be used for analysis, visualization, and storage. Now, our algorithm receives in input an image, identifies the color marker in the image, and then computes the marker average color intensities. The markers are not characterized by a precise shape, size, and position because these topology information are identified by the algorithm itself. Now I'm going to go through the step of the algorithm. At first, we have the region of interest, ROI, extraction. So we have an input image of our test, and we want to extract only the region of interest containing the marker. We don't know what is the size of the marker, what is the shape, but we know that the marker is there. To do this, in our approach, we have used April tag on corners. This enables also the use of the algorithm on smartphones. Of course, other methods are available. In our approach, the idea is to put some little marker on the edges, on the strips of the real sampling test. We identify these tags, they look like small QR codes, and therefore we know what are the corners of the strip. Now by doing rotation, the warping and cropping, we can get out our region of interest on which the algorithm can work upon. The next step is threshold calculation. So on a black and white version of the image that we get, we run an histogram. We count what is the amount of pixel of each color. And if there is a color marker, we will end up with two peaks. One peak is the background color, one peak is the foreground color. And we select a threshold, a value that it is in between these two peaks. We then use the threshold in order to generate a binary mask. So whatever pixel is above this threshold becomes white, whatever pixel is below this threshold becomes black, therefore creating a certain mask on which we can run contours detection algorithms, getting out a lot of contours. Some of these contours will be the contours that are identifying our marker. Some of them will be contours of noise or other impurities in the picture, like dust. We run this through a process called color contour filtration, on which we try to identify what are the contours of our markers. We have different filtration techniques which we have developed and test on real sample. So we filter contours that do not contain specified points. We filter by area. Usually dust and noise have very small area. We can filter contours that touch or not touch edges of the pictures. In this case, we may be able to identify strips or dots or whatever shape we want. We filter by convexity defects and by real expected centroid difference. Now on the paper there is the description in detail of all this kind of filtration. Here I'm just to give you an idea, I'm gonna describe the one by convexity defects. So we have noticed that the contours of the real marker are quite regular, while contours of noise or markers that are not real, for example dust, they are very curly. So what we do is to measure a parameter called convexity defects, which is the depth between our contour and an envelope of the contour. By putting some limits, some constraints on this value, the convexity defect depth, we can select some contours and discard others. At this point, we should have ended up with a set of contours. If not, then we run a deep search, which runs only if no contour is found. This is a more thorough search of the contours using different values of the threshold and different parameter for the filtrations, and takes longer time, so is more computationally heavy, but it is able to better identify contours of the marker. Once we end up with a set of contours, for example here the ones in yellow, we go through a step called hierarchical selection. And this is quite an easy step because we just select the most outer or the most inner contour. We do this because experiments have shown that a coffee ring effect is sometimes present. In this case, we will have two contours, one inner and one outer identified. And depending on the application, I may want to average the pixel color, including or not the coffee ring. Finally, we can do the average of the pixels that are inside the contours, which are the ones that belong to our color marker, and the result translation. The average is weighted, so I can give more weight to certain pixels than others. In this case, if we include the coffee ring, we may say that those pixels count less in the average, and the result translation converts the result, which is usually as RGB, red, green, blue values, into something that a user can use, for example, bacteria concentration values. Now we move on with the experimental evaluation. This is our digitalization device, the first prototype we built, and it consists of a computing device, which in our case is a Raspberry Pi 2, a digitalization box, and here you can see the readout, which can be inserted into the digitalization box with a little drawer. The algorithm was implemented in Python 3 and uses functions provided by OpenCV for basic image processing. Inside the digitalization box, we have light reflectors and 4 white LEDs. And the purpose of this is to provide diffused uniform lighting of the sample. And on the top, on the lid, we have a 5 megapixel camera equipped with a macro lens, which is focused exactly at the distance where the lateral flow assay strip is to be found. So we can take very nice pictures of the strip. Here you can see the algorithm in action with different markers. These markers are artificial. That means that are not created by real bacteria, but they are we just used a stain to create them. On the first row, you can see an uniform round marker, which is quite easily identifiable also by the naked eye. And here we get the result of our algorithm. We can see the contour which is identified and the value of RGB. Here we have a stripe marker, which is equivalent to the top one. On the third row, we have a challenging round marker. Now, this is almost not visible here in the picture in the presentation, but our algorithm was able to identify the contours and say what is the value of the RGB, of the average of the pixel inside the contour. And for this one, we actually had to run the deep search. So to find the first two, it was quite quick. To find the last one, we need to run the deep search. Finally, we also tested with an empty region of interest. In case that there is no marker, the algorithm should be able to say there is no marker and not identify some artifacts as the marker. We also tested with real sample. These are created using real bacteria, and so it's the real product. And we can see that also in this case, we are able to identify. In this picture, we can notice that there is a coffee ring, which is clear actually, and we only take into account the inner part. So this is where the hierarchical selection step of the algorithm plays a role. Also in this case, for the real strip, we tried with a region with no marker, and the algorithm is able to identify that there is no marker, even if there is some specks of dust. We evaluated our algorithm also in terms of execution time. Here in this table, you can see the result for 3 different type of markers: uniform round, challenging round, and empty, so no marker. And we can see that a good percentage is the region of interest extraction, which is carried out in our case using April tag. So some of the time is actually spent just to identify where the April tags, the corner of the strips, are, and to crop the warp and rotate the image. And then another good amount of the time, good percentage, is the deep search when it runs. For example, here on the uniform round, it is 0 because the deep search did not run. It was quite easy for the algorithm to identify the marker. But for the challenging round region, the deep search takes 700 milliseconds. The same applies for the empty region, where it takes around 300 milliseconds to identify that there is no marker. If we look at the total, all the results are sub-seconds, so the detection itself is quite fast. So now we have a little summary of the contributions of this work. What we presented was a computer vision algorithm which is used for the digitalization of colorimetric lateral flow assay readouts. We used it in our project to identify the concentration of bacteria, but in principle it should be able to identify any concentration because it just reports the color of the marker. Our algorithm is quite special because it is not based on a predefined position, shape, and size, but it is able to identify the marker at runtime independently by the characteristic of the market, and it uses 2 levels of search, fast and coarse, and if this fails, it runs in a more exhaustive deep search, which is triggered when challenging markers are present. We have evaluated and used this work both with artificial and real samples in the lab, and overall the execution time is quite fast, less than 1 second on a Raspberry Pi 2. This concludes my presentation. Thank you for your attention.